Hello everybody. I'm going here to discuss about first nitrogen fixation and after that biological nitrogen fixation. So, what is nitrogen fixation? Nitrogen is, as we know, present in many forms in the atmosphere. The atmosphere contains about 78% by volume this molecular nitrogen. And this nitrogen actually is taken up by the plants by breaking the stable triple bond in these two nitrogen atoms, which then are converted into either ammonia or nitrate ions. So plant can uptake these two forms. It cannot uptake this form, inert form, because plant is unable to convert directly this nitrogen into the ammonia or nitrate. So the conversion of this nitrogen into ammonium or nitrate ion is called nitrogen fixation. So this is nitrogen fixation. So what are the types of this nitrogen fixation? It can be biological or non-biological nitrogen fixation. Nitrogen fixation. In this case, we require microorganisms, that is usually prokaryotes like bacteria, which fix the nitrogen into NH4 ions. So we require here the microorganisms, and 90% of the nitrogen fixation is by this method. But in non-biological cases, photochemical or lightning reactions are involved. So microorganisms are not involved here, and 90% is by biological nitrogen fixation and rest is by these methods. So this is biological and non-biological method. Biological nitrogen fixation, it's a reductive process. That means nitrogen gets reduced here by the use of electrons. Two kinds of reaction can be there. First, nitrogen can be converted into this ammonium ion plus hydrogen, two ammonium ions and one H2 molecule by using 8 electrons. So if it use only 6 electrons, we cannot get this hydrogen atom. We can get only NH3 molecules, are 2 in number. So these are the two reaction types which we can get. After that, we shall first discuss here about the nitrogen fixing bacteria, which can fix the atmospheric nitrogen for plants into ammonium ions. So they can be free living or they can be symbiotic. In symbiotic case, they keep themselves in symbiotic relationship with the host cells or the host tissue in order to take the nutrition for the survival. In free living, they can be anaerobic or aerobic. The both aerobic as well as anaerobic can be photosynthetic as well as non-photosynthetic. So anaerobic photosynthetic type free living nitrogen fixing bacteria, example is chromatium, and non-photosynthetic is clostridium. In case of aerobic photosynthetic type, the example is blue-green algae, and non-photosynthetic type azotobacter. We shall discuss here about mainly symbiotic type. There can be two cases. The microorganisms or nitrogen fixing bacteria can form the root nodules, or they can fix the nitrogen, but they cannot form the nodule. For example, in Anabaena azole, it keep symbiotic relationship. It maintains the symbiotic relationship with the water fern azolla, but it is not able to form the root. It is not able to form actually the nodule. In case of nodule, mainly leguminous plants, the nodule can be formed in the roots or on the stem, like root nodule or stem nodule. Mainly, the example rhizobium is here, which can form root nodules in leguminous plants. So, we are discussing here about the rhizobium, its root nodule formation in the plant. So, what is the mechanism of this root nodule formation in the leguminous plants by rhizobium species? First, this rhizobium species is gram-negative bacteria. It can be free living as well as it can be symbiotic, and it's rod shaped soil bacteria. So, in order to start the mechanism of root nodule formation, we require certain genes that are called as nodule forming genes. And these nodule forming genes can be of two types: nodulin genes, which are actually present in the host cells, or nodulation genes, which are produced by the bacterial cells. Nodulation genes can be common nodulation genes, or they can be host specific nodulation genes. Common nodulation genes are nod A, B, and C. These are called common because they are commonly present in all the strains. However, these host specific nodulation genes are present differently in the strains. The importance here of these common nodulation genes is that they produce the products. These genes produce the products which form the enzymes, and these enzymes are responsible for the formation of nod factors, or we can say nodulation factors. These nod factors induce these genes, nodulin genes, in the host cells, and they cause the elongation and curling of the root hair. When they cause the elongation and curling of the root hair, when they get entry into the cortical cells, they act as mitogen and cause the division in these cortical cells. As a result, the cortical cell number gets increased very much. So they form nodule primordium, which ultimately results into the formation of root nodule. So, what is the chemical nature of these nod factors? These nod factors are composed of lipocyto-oligosaccharides. Most important thing, lipocyto-oligosaccharides, that are formed of lipids, that's fatty acids, or carbohydrates, oligosaccharides, which are formed of monosaccharides, 8 to 10 monosaccharides. These monosaccharides are N-acetyl D-glucosamine, which are linked up by beta-1,4 glycosidic bond. Actually, these lipocyto-oligosaccharides are chitin derivatives. So, if we start the mechanism here, the plant cells secrete metabolites like flavonide, isoflavonide, homoserine, and these are called the chemotactic substances. And on the other hand side, these bacterial cells release NOD factor. This NOD has the importance that it recognizes the site where the flavonide, isoflavonide, or homoserine has been released. So when it recognizes the site, it returns to the bacterial cells, and here induce the expression of nod A, B, C common nodulation genes. And the function of these common nodulation genes is what? They form the nodulation factors, which ultimately lead the elongation and the curling of the root hairs. So, the nod A, which is the product of nod A gene, is an N-acetyl transferase which catalyzes what? Addition of fatty acyl chain. Then nod B, which is chitin oligosaccharide deacetylase, it removes acetyl group simply from the terminal nod reducing sugar. And finally, nod C, its chitin oligosaccharide synthase, it links N-acetyl D-glucosamine monomers. So after that, we can see here an important kind of protein, ricadicin protein, is here, which is present on the surface of the bacterial cell. It is very important because it acts as calcium binding protein, and it is used for the attachment of bacteria, that's rhizobia, with the host cell. Once the ricadicin protein bind with the bacterial cell wall, plant cell wall, these bacteria, these rhizobia, degrades the external cell wall and elongates this plasma membrane. Actually, this elongation of this plasma membrane is due to the fusion of Golgi vesicles with the plasma membrane. And this elongation, this thread, is called as infection thread, which is formed by lipid bilayer. And after the formation of infection thread, it gets elongated. So when these continuously dividing rhizobia reach to the cortical cell surface, invagination of the plasma membrane will take place. After invagination, the bacteria detach from the plasma membrane, and certain kind of structures are formed, which are called as circular bacterioid, which contain rhizobia, groups of rhizobia. So these bacterioids are surrounded by the membrane, which is called as peribacterioid membrane. Here the case arises as it is the anaerobic process. We know that the cell is containing the oxygen, but here the nitrogenous enzyme, which is responsible for the reduction of this nitrogen into the ammonium ions, and then it is inactivated by oxygenase. Mainly its nitrogenous enzyme actually composed of two enzymes, denitrogenase reductase and denitrogenase. This denitrogenase is inactivated in the presence of oxygen. So what the bacterioid or what the rhizobia do and what the plant cell do, the rhizobia produce heme group and cell produce the globin group, and this heme group is actually transported to the cytosol. It is present in the cytosol, not inside the bacterioid membrane. And this heme group plus globin, these two components form leg hemoglobin. And this molecule leg hemoglobin, which is present in the cytoplasm, and oxygen is also present in the cytoplasm, what this do, it is having the higher affinity towards the oxygen. It bind with the oxygen, form oxy-leg hemoglobin. As a result, the concentration of oxygen will get decreased in this particular cell. So the nitrogenous enzyme is wholly active now, mainly denitrogenase. As this nitrogenous enzyme is activated now, it reduces the nitrogen into the ammonium form. Okay, so the nitrogen has been reduced into the ammonium form, and our nitrogen fixation step has been completed. But at last, when it is completed, this, as it is present in the higher concentration, ammonia ions are toxic to the plant cells. So these cells convert these ammonia into certain other molecules, that's converted into amides, that's asparagine or glutamate or certain other molecules. But here one important thing I forgot to say, these are NIF genes. NIF genes are also important. NIF genes are required for the formation of this nitrogenase enzyme and for catalytic process of nitrogen. And we know this biological nitrogen fixation is a reductive process, and they require electrons. And where from they get the electrons? The electrons are donated by reduced ferredoxin. Important point here, the electron donor here is reduced ferredoxin. So in total 16 ATP molecules are required to reduce one molecule of nitrogen. So this was all about the nitrogen fixation and biological nitrogen fixation. Thank you so much.